So, good evening to all. Welcome to week 8 tutorial session of experimental biotechnology. So, I'm the teaching assistant. I'm Ananya Chatterjee, PhD student in IISc Bangalore, and the course instructor of this course is Professor Vishal Trivedi from IIT Guwahati. So, today we are at week 8 tutorial session. So far we have completed 7 week tutorials, and also I discuss every assignments which you get from these courses. So far till assignment 6 we have discussed, and also along with the live assignments we also discuss previous year's assignments, so that it will be helpful for you to solve this year's live assignments. And every day's tutorial sessions we record and upload in this YouTube link, and all the lecture slides of everyday's tutorial sessions is getting uploaded in this folder link. So if you have any doubt from previous days and previous tutorial sessions or any assignments, you can let me know. Else I will start with week 7 assignments, problem solving session. So the first question was in week 7 assignment is that which of the following good sorry, good evening, Trishala. I started teaching, but my mic was I was muted. Okay, I will start. I think you could not hear me. Yeah. So welcome to today's class. So far we have completed till week 7, so today's is week 8 tutorial session, and also among assignments we have discussed so far till assignment 6. So today I will be discussing week 7 assignments which you had previous week, and also today we will be discussing week 8 week 9 assignments which from the previous year. Okay, so that this year's week 9 assignment is live, so that you will get benefited by getting a sense of how what are the questions you can maybe ask. Okay, if you have any doubt from previous class or any assignments and all, please feel free to let me know. Else I will start with this discussion of this problem, whatever you were given. Okay, so let's start. I think others will also join soon. Let's start with this problem solving. The first question was which of the following is involved in humoral mediated immunity. Okay, number 1 T cell, number 2 B cells, antibody, plasma B cells. So humoral mediated immunity is directly involvement of antibody. Okay, we know that B cell produces antibody, and antibody is the one which is directly involved in this humoral mediated immunity, and cell mediated immunity involvement is there of T cell. Okay, for cell mediated immunity. So the answer will be for humoral mediated immunity, it will be antibody. So the Y-shaped structure of an antibody is composed of how many protein chains? Okay, so that we know that the heavy chain and light chain. Okay, heavy chain and light chain. So it's composed of how many protein chains? It's composed of 4 protein chains. So the answer will be 4. In which phase of the antibody production there is entry of pathogen in body and contact with immune cells? So where in the antibody production only pathogen enters in the body and contact the immune cells? So the curve is like that. So this is the curve where this is the lag phase. Okay, so this is the lag phase where pathogens enter inside our body, and then they start our antibody is getting produced in this log phase. So the answer will be entry of pathogen in body occurs in the lag phase. Which of the following is antigen presenting cells? Macrophages, T cells, activated B cells, plasma B cells. So antigen presenting cells, like those are the cells which presents antibody through receptors to MHC1, MHC2 complexes. So like through MHC1, MHC2 complexes, those are examples is macrophages. Okay, macrophages, dendritic cells, those are antigen presenting cells. How are monoclonal antibodies produced? Options: by collecting antibodies from different animals. No. By stimulating a single B cell cloning and fusing it myeloma cells. This is the correct answer. Or other options are by isolating antibodies from a mixture of different B cell clone. No. By cloning T cells and generating hybridomers. No. So the answer is by stimulating only single B cell. So that is why it is called monoclonal antibodies, because it is stimulating only B cell clone and using myeloma cells so that the amount of cell division remains infinite. Okay. So HAT medium inhibits nucleic acid synthesis, cell membrane synthesis, antibody synthesis, protein synthesis. So HAT medium inhibits nucleic acid synthesis. We discussed in the previous class regarding those myeloma and hybridoma technology. So HAT medium inhibits nucleic acid synthesis. Which of the following is commonly used as a carrier particle in the indirect hemagglutination reaction? So the carrier particle in the indirect hemagglutination reaction is red blood cells or RBCs. Which of the following disease can be diagnosed using the indirect hemagglutination reaction? So influenza, bacterial infections, malaria, Lyme disease. So we have discussed that in the case of detection of influenza, we because of those salicylic acid interaction, we can diagnose influenza by using indirect hemagglutination reaction. The outer colony double immunodiffusion technique is used to determine blood type, detect specific antigen in a sample, quantify antibody concentration, purify the antigens. So the outer Luni double immunodiffusion technique is used to detect specific antigen in a sample. Which parameter can be estimated by measuring the diameter of the precipitin ring in the radial immunodiffusion assay? So which parameter can be estimated by measuring the diameter of the precipitin ring? Options are antibody concentration, pH of the solution, viscosity of the gel, antigen concentration. So in the case of radial immunoassay, we know that the diameter of the precipitated ring we can estimate to understand the antigen concentration. The bait molecule used in pull down assay is typically a small molecule drug, an antibody, protein of interest, DNA plasmid. So the bait molecule is a protein of interest. Next question. The target molecule in a sandwich ELISA is captured between two antibodies. So what are those antibodies? Primary antibody and enzyme conjugated secondary antibody, blocking antibody and substrate conjugated antibody, capture antibody and detection antibody, secondary antibody and tertiary antibody. The answer will be capture antibody and detection antibody. How does competitive ELISA work? So options are: the target antigen competes with a labeled antigen for binding to limited antibodies. The target antibody competes with a labeled antigen for binding to limited antigens. The target antibody directly binds to enzyme. The target antigen directly binds to substrate. So it C and D option will never be true. It doesn't bind directly. So there should be a competition. So competition between the target antigen. So what happens? The target antigen competes with the labeled antigen. Okay, for binding to limited antibodies. So antibodies are limited in this case, and target antigen competes with another labeled antigens for binding to those limited antibodies. What is the purpose of membrane blocking in Western blot? Options: to prevent proteins from binding to the membrane, no. To remove excess antibodies, no. Prevent non-specific binding and reduce background noise, yes. So the answer will be prevent non-specific binding and reduce background noise. And the reagent used to develop HRP based secondary antibody system in Western blot is options are BCIP-NBT, substitute 1,2-dioxetane phosphate, diamidazole, luminol H2O2. So HRP based secondary antibody, we have read that luminol H2O2 is a reagent which we use to develop. So next we will be discussing week 9 previous year assignment, but before that we will discuss week 9 contents, okay, so that we'll come back and solve the assignments clearly. So in the week 9 content there is cell biology part 2. So we were discussing different types of cells and then different types of cell media. Today we will be starting discussing about microscopy. So microscopy is a tool that are used to observe the small organisms or object and even macromolecules. Okay, so we use this technique to observe small organisms, cells and different macromolecules. So the instrumentation behind microscopy is, so here the light is produced by a lamp with tungsten filament as a source. This is a light microscope example, okay, as a source, and light rays are focused on the sample by the condenser. The specimen is kept on the stage and formed by clip present on the side. The light diffracted by the sample is then collected by the objective lens. So objective lens can be varied from 10x to 100x magnification in the case of simple light microscope, and additional magnification is achieved by the eyepiece, usually gives additional 10x magnification. So this is objective lens just before the sample, and this is the eyepiece or ocular lens. So there are two variants, one is upright, another one is inverted. So in upright, this objective lens are just above the sample, and this inverted, the objective lens are below the sample. So this is inverted, that's why its name is inverted. So in an upright microscope, the objective turret is usually fixed and the image is focused by moving the sample stage up and down, okay. And in an inverted microscope, the sample stage is fixed and the objective turret is moved up and down to focus the final image. Now fluorescence microscopy. So what happens in fluorescence microscopy? In an epifluorescence microscope, the illumination of the specimen as well as the collection of the fluorescence light is achieved by a single lens. This has become possible due to incorporation of dichroic mirror in the optics. So a dichroic mirror is largely reflective for the light below a threshold wavelength and transmissive for the light above that wavelength. So immunofluorescence microscopic analysis of cell surface markers is straightforward, wherein the cells are treated with fluorescently labeled antibodies and studied under microscope. So this is a broadband light source, the excitation filter, the sample, emission filter. So emission filter is there, and then this is the dichroic mirror. So from sample light, so light passes through. So what happens? From a light source, light comes through excitation filter, and then by reflection through the dichroic mirror, it comes into the sample, and then after diffraction it goes through the emission filter and falls on the CCD camera. So by fluorescently labeling any macromolecule, you can do immunolocalization by using so immunolocalization you can do by specifically using any antibody against specific proteins. So this is immunolocalization. So what happens? Stop the movement of biological macromolecules fixations. At first you have to fix the biological macromolecules. Wait for antibody to enter inside cells. This is permeabilization. So you need to permeabilize your cell so that antibody gets entered. Hide the non-antigenic sites. You need to block some to avoid some non-specific interaction. Observation. So you need to mount on the microscope and then observe. Okay, so just hold on for 5 minutes. I'll be back. So we'll be discussing immunolocalization. So what are the materials required for immunolocalization? So we need methanol, acetone, PBS 1x, 1% Triton X100, BSA, fat-free acetylated, primary antibody, which is against antigen, secondary antibody, and epifluorescence microscope. So BSA, we have to prepare 5% BSA solution in PBS and filter with 0.45 micron filter to remove particulate matter. Okay. Primary antibody. So this is the antibody which can be developed against a protein which is antigen of interest in rabbit or mice. Secondary antibody, an antibody coupled with fluorescent marker such as FITC and directed against mouse IgG. And we also need a epifluorescence microscope. So these are the procedures of immunolocalization. So what is that? At first we need to fix our sample. So we need to do fixation. So this is the first steps and it is required for two purposes. The first one is stopping any biological activity, and second, it stops the relative movement of cellular components and intracellular macromolecules. In addition, it also reduces the damage of the cell membrane and then morphology. And so for fixation of the sample you need methanol, acetone, and you need to hydrate the sample with 1x PBS also. After that you need permeabilization. So the membrane is generally non-permeable, so that's why you need to do the permeabilization. For that you can add 1% Titan X at room temperature, and you can permeabilize the cells. So the cells will be at first like that, and then what will do, you will do the permeabilization so that your dyes and then antibodies can enter into the cell. So cell membrane is non-permeable to the charged as well as macromolecules, so only small molecules or hydrophobic dyes can enter. So for that reason cellular membrane needs to make porous by particularly removing lipids from them. So this process is known as permeabilization. So cells are permeabilized with 1% Triton X100 for 15 minutes at room temperature. Now blocking. The intracellular spaces contain several antigenic sites, and this need to block to reduce non-specific binding of the primary antibody. The cells are incubated with 5% BSA in 1x PBS for 15 minutes at room temperature. This step will allow masking on non-specific antigenic sites. Primary staining. Incubate the sample with primary antibody 1:15 2% BSA for overnight at 4 degrees Celsius or 1 hour 37 degree. The primary staining at low temperature reduces the background signal and give good staining for sample, whereas staining at room temperature gives more amount of non-specific signal. So that's why the primary staining at room temperature reduces the background signal and give good staining, whereas staining at room temperature gives more amount of non-specific signal as well. So wash is needed. The primary antibody needs to be washed to reduce the background signal. So sample is washed in 2% BSA prepared in PBS. After that, what we need to do, we need to stain with secondary antibody. So we need to incubate the sample with secondary antibody. It can be dependent on the how much dilution, dependent on the concentration of the secondary antibody. So for overnight, or also you can do 1 hour 37 degree. Washing. The secondary antibody needs to wash with to reduce the background signal. Sample is washed with 2% BSA prepared in PBS. Mounting. The sample is sensitive to the loss of water and needs to preserve in a mounting media containing glycerol. In addition, fluorescence signal is sensitive to the high laser beam and it's required protection by adding anti-fading agent. In a typical mounting media, glycerol containing PPD, which is paraphenylene diamine, is used to mount fluorescent sample. So you need to remember this required media, the mounting media, where glycerol containing PPD, that is paraphenyl diamine, to use to mount fluorescent sample. So observation and visualization. Sample is fixed on the microscope stage. So sample is fixed on the microscope stage and then observe under bright light to check the cell's morphology by turning focusing knob. If the sample's morphology is good, then it can observe under fluorescence channel. So this is the observations and visualization of sample. So what happens, we need to fix the sample in the microscope. Please ignore the background noise because of Ganesh Puja. And happy Ganesh Chaturthi to all. So we need to fix our samples on the microscope stage and then observe under bright light to check the cell's morphology by turning focusing knob. If the sample's morphology is good, then it can observe under fluorescence channel. Okay. So here again the same materials they have used to check. So there one particular cells has been used, and those are cultured in DMEM media containing 10% FBS and 1% antibiotics. So they have removed the cells from the cell culture plate by trypsinization, then plate around 10,000 cells on a 12 millimeter cover glass, incubate it in the 24 well dish with 0.5 ml DMEM media containing FBS and antibiotic cocktail. So incubate cells overnight at 37 degree, 5% CO2, and it will allow the cells to attach the cover glasses. Wash the cells with DMEM without FBS media. Prepare a suspension of latex beads in DMEM without FBS media. Remove media and add bead suspension to the well. Centrifuge the 24 well at 1000 rpm for 1 minute. Incubate the plate for 1 hour at 37 degree, 5% CO2. Wash the well with 1 ml DMEM without FBS media to remove uninternalized beads. Fix the biological sample with methanol. As methanol is to acetone 7:3 mixture. Stain the cells with Philippine for 1 hour. Keep 1 drop of mounting media, which is glycerol mounting media containing anti-fading agent, on the glass slide and keep the cover glass on it. Form the cover glass by making a thick film by nail polish. So typical phagocytosis of beads with represented by the appearance of the beads in the face, and some bead will be circled by in this image. So this is how it will look under epifluorescence microscope. Okay. Now we'll be discussing about resolution. So the term resolution is the minimum distance at which two distinct points of a sample can be separated. Okay. So the resolution of a microscope is linked to the numerical aperture as well as the wavelength of the light used to examine a specimen. So resolution dependent is dependent on the wavelength and numerical aperture. So the formula is D equals to lambda by 2 nm. If using a green light of wavelength of 540 nanometer and an oil immersion objective with a numerical aperture of 1.45, then the limit of resolution will be 177 nanometer. So this is by using the formula you can calculate this, that wavelength is given, numerical aperture is given, so what is the limit of resolution. Okay, and this is the electromagnetic wavelength series where you can see the different wavelengths of different electromagnetic rays. So what is electron microscopy? So there are two basic models of electron microscope. One is scanning electron microscope, another one is transmission electron microscope. In a scanning electron microscope, the secondary electrons produced by the specimen are detected to generate an image that contains topological features of the specimen. The image in a TEM, on the other hand, is generated by the electrons that have transmitted through a thin specimen. So this is a anatomy of scanning electron microscope. So here what happens, the focused beam of electrons is scanned across the surface in a raster fashion. Then this scanning is achieved by moving the electron beam across the specimen surface by using by using reflection or scanning coils, the number of secondary electrons produced by the specimen at each scanned point are plotted to give a two-dimensional image. Week 9 assignment will be discussing after discussing this. Okay, so the scanning electron microscope, most electron microscope have the detectors for the secondary electrons and the backscattered electrons. So the backscattered electrons can provide useful information in this case about the composition of the sample, and materials with higher atomic number produce brighter images. So here you can see this is the electron gun, and then these are the scanning coil, this is condenser lens, then this is the specimen and this specimen holder. So after light gets fall onto the sample, it gets deflected and then it comes into the contact of detector, and from that it goes to photo multiplier and then the detection system. So if you want to visualize something, so the materials you require: ethanol, foil, some specimen, gold spotter, scanning electron microscope and desiccator. Procedure: you have to make sample. So the cells or non-biological material for same analysis needs to place on a small piece of aluminum foil. After that you would need to fix. So biological samples are fragile and fixation of biological sample is required for two purposes: stopping biological activity and stop relative movement of specimen. So we need to fix samples using 2 to 5% glutaraldehyde and 1% osmium tetroxide in 50 millimolar sodium cacodide dilute pH 7.4 for overnight. Procedure: dehydration. So biological samples are fragile and contains large amount of water. Water present in the biological sample diffract electron rays and may increase the background signal. Following osmium fixation, water is chemically extracted from the specimen using a graded series of ethanol. So it's performed in the following steps: sample is incubated with 50% ethanol for 30 minutes, sample is incubated with 70% ethanol for 30 minutes, and then sample is incubated with 80% ethanol for for 30 minutes, again 90% ethanol for 30 minutes, 95% followed by 100%. So sample is incubated with anhydrous ethanol for 30 minutes. So like that, slowly gradually you have to take out all the water inside the sample. Then step 5 is sample coating. So the specimen needs to be coated with the conductive material to view with scanning electron microscope. In the sputter, a discharge is formed between anode and cathode using a suitable gas. The bombardment of gas ions on cathode material, usually gold, erode the target material and deposit it on the specimen. The target used in the sputter consists of 60% gold and 40% palladium. The uniform deposition of sputtered material form even coating on the surface of specimen and make the specimen conductive to perform scanning electron microscope. Step 4 is drying. So during dehydration, samples needs to prevent air drying. Once the dried material is removed, it needs to be stored in a desiccated environment until viewing. Okay. So this is one image of E. coli cells. So this is the overall like the surface view of cells you can see. So this is the image taken from a scanning electron microscope. Now we'll discuss about transmission electron microscope. The image like line or the anatomy is very much same. Only the difference is the detection of which electron we are detecting. So the electrons which are transmitting the sample, we will be detecting those. So in comparison to light microscope, TEM is in, so we don't have any bulb or for the light source, we have electron gun. Okay, so electrons are the so we don't have light, basically light rays, we have electron rays. Again we have condenser lens, we have specimen holder specimen, we have projection lens. So this is the ray diagram or the like the microscope anatomy for transmission electron microscope. So this usually have thermionic emission guns and electrons are accelerated anywhere between 40 to 200 kV potential. However, TEM with greater than 1000 kV acceleration potentials have been developed for obtaining higher resolution. Owing to their brightness and very fine electron beams, field emission guns are becoming more popular to the electron guns. Transmission electron microscope stop the movement of biological macromolecules. You have to fix, you have to remove water, dehydration, make a hard block for cutting, embedding. So if you so for a cell or tissue section, if you want to cut, so you need to do microtomy for like sectioning. So for that you need to embed the sample into hard block so that your cutting thin sections become easier. So thin section of around 10 to 80 nanometer. So you will be doing sectioning after that you have to hide the non-antigenic sites, so block, and then staining with antibodies, and followed by staining with contrast reagents. Okay, so the materials at first paraformaldehyde and glutaraldehyde, PBX 1x2 BSA, primary antibody, secondary antibody, and antibody coupled with gold particle, uranyl acetate as a stain, osmium tetroxide as contrast, propylene oxide, ethanol, epon resin, white resin, microtome, and transmission electron microscope. So these are the requirement for microtomy along with transmission visualization the sample under transmission electron microscope. Procedure. Fixation. The sample for TEM are fixed by two different ways, immersion or perfusion. So fixation time, concentration of fixative agents depends on tissue thickness. It is performed in following steps. Sample is incubated with 2% paraformaldehyde or 2.5% glutaraldehyde in 50 millimolar sodium cacodiol pH 7.4 overnight at 4 degrees Celsius. Post fixation, samples are incubated with 2% osmium tetroxide in 50 millimolar sodium cacodiol pH 7.4 for overnight 4 degrees Celsius. Now you have to follow dehydration procedures. Then what you will do, embedding. You have to embed a thin 10 nanometer to 100 nanometer section of the sample so that the electron can pass through it to form an image. Biological samples are fragile and cannot be processed to cut thin sections, hence sample needs to be embedded in a solid matrix to cut the section. So EPON, there is one process is called EPON embedding. To remove the solution and sample is incubated with propylene oxide and EPON resin 1:1 overnight. Remove the solution and sample is incubated with fresh EPON 812 resin for additional 1 to 5 hours. Dispense few microliter of fresh EPON 812 resin in polyethylene capsules and specimen is transferred into it. After that, place capsule at 60 degrees Celsius in hot oven for 48 hours. Ehler white embedding. Dehydrate the specimen in ethanol as described. Incubate tissue or cells for 30 minutes in a mixture of ethanol and Ehler white resin. Subsequently, incubate tissue or cells for 30 minutes in mixture of ethanol and Ehler white resin 2:1. Incubate tissue or cells in Ehler white resin for 1 hour. Repeat this step 3 times. Finally, leave the tissue or cells for overnight in 100% Ehler white resin. Fill the capsule with Ehler white containing tissue and cell and seal the capsule to allow polymerization of resin in 50 degrees Celsius oven overnight. So after that you can proceed with sectioning. Initially, thick sections are cut with the help of razor blade to reach the tissue in the block. Afterwards, the block is mounted on the microtome and then the ultra thin sections are cut and floated on the water placed within the boat. Cut sections are stacked on each other in a definite pattern known as ribbon. These ribbons are collected on electron microscope grid. Staining to visualize cell structure. Incubate the grid in uranyl acetate for 2 hours. Subsequently in lead citrate for additional 2 hours. Uranyl acetate staining will allow to observe the cellular structure and to study the changes in the cellular or subcellular morphology. You can also do immunogold leveling. So you have to then, what you have to do, you can directly visualize at this stage, but if you want to be sure whether your macromolecule of interest is present, you can do immunogold leveling. So you have to block the grid sample with 5% BSA for 45 minutes at room temperature. Incubate the grid in the primary antibody for 1 hour at room temperature or overnight at 4 degrees Celsius in humidified petri dish. It is advisable to centrifuge primary antibody on full speed for 5 minutes to remove aggregated antibody. Dip the grid in PBS, drop 3 times. Dip the grid in PBS containing 2 drop for 6 times. Incubate the grid in the secondary antibody coupled to 10 nanometer gold nanoparticle for 1 hour at room temperature or overnight at 4 degree in a humidified petri dish. Dip the grid in drop of PBS containing 2 for 6 times again. This is a wash step. Again you have to drip in PBS only for 3 times. Finally, stain the grid again with uranyl acetate for 1 minute and lead acetate for 30 seconds, and you can visualize. So this is one image of immunogold labeling imaging. So these are the antibodies with gold particles bound with the sample. Now we'll discuss one research problem. So scientists have discovered a new bioactive phytochemicals from neem tree, and now he wants to test this effect on propagation of malaria parasite. So bioactive phytochemicals, it's inhibition of malaria parasite propagation. Okay. So malaria parasite life cycle is that you have a trophozoite stage, and then zygones, then merozoite ring stage, and this is the life cycle of malaria parasite inside human. So experimental data, what they can do: Plasmodium falciparum synchronized to ring stage, incubated with increasing drug concentration. Those Plasmodium falciparum are incubated with increasing drug concentration. Then smear after 35 hours to study side joint maturation. Smear after 72 hours to study the effect of drugs on RBC reinvention, drug removal. And smear after 72 hours to study static or sidal nature of drugs. What are the materials you need? RPMI 1640 cell culture media, Albumex 2 0.22 micron membrane filter, filtration unit, autoclave, vacuum plug, pump, upright microscope. Procedure. Step 1, preparation of ring stage of malaria parasite. This is the first step where parasite culture mixture of stages of malaria parasite is synchronized to the ring parasite containing RBCs. It has following steps. Take 4 ml of a culture of greater than 5% parasite. Centrifuge the parasite culture at 720 G for pellet down. Resuspend the parasite pellet for with 4 ml of 5% sorbitol and incubate for 10 minutes at room temperature. Mix and shake it 2 to 3 times. Centrifuge the culture at 720 G and wash it 3 times with media and bring the parasite to the 5% hematocrit. Repeat the step 1 to 4 after culturing the parasite after one cycle, approximately 48 hours. Calculate the parasitemia after gym sustaining, and calculation of parasitemia is discussed later. So preparation of compound solution. For this, the test compound can be dissolved in the organic solvent at concentration of 5 mg per ml. It is recommended to use DMSO as solvent, has no significant effect on parasite growth. Setup assay. Parasite culture synchronized at ring stage by desorbital treatment brought to the 1% parasitemia with 3% hematocrit. In a total volume of 100 microliter, 50 microliter parasite culture is mixed with the various concentration of test compound in a 25 microliter and remaining complete media. Chloroquine can be added as positive control and solvent as a negative control. Incubate the compounds for 48 hours. Monitor the appearance of hemolysis or any such effect. If appears, stop the assay and screen the compound using other assay. Monitoring the growth of parasite. After 48 hours after exposure, smears were made. Parasitemia has been determined. Then you have to calculate IC50. The number of side joint containing RBCs were counted against each concentration. So the side joint inhibition data from the in vitro side joint inhibition assays of the above compounds were fed into a specially pre-programmed Excel sheet. To determine the nature of action, parasitostatic or parasidial, in 100 microliter volume, 3% hematocrit with 1% parasites were exposed to trial compounds for 48 hours. After 48 hours water done, parasites will be washed twice, okay, with complete media, and then again you have to incubate for around 48 hours in drug-free media. Okay, then you can make smears and parasitemia has been determined microscopically. So next experiment is that scientists are routinely propagating mammalian cells, and he has observed a new medium for cell proliferation. He has developed. Okay, now he wants to design the experiment to count number of viable cells and dead cells. So how you can do? You can use trypan blue. Okay, so trypan blue is basically a charged dye, and viable cells exclude this dye due to the presence of membrane potential, where dead cells, in the absence of membrane potential, accumulate the dye in the cytosol. So trypan blue is only permeable in the case of dead cells. So what we can do, this is a hemocytometer. We can place some amount of cells, around 10 microliter or 8 microliter cells, onto this hemocytometer, and we can count. Whatever cells are blue, those are dead cells, and whatever cells are white or transparent, those are live cells. So for this also we need glass slide, cover slip, inverted microscope with faceplate, and hemocytometer. So for that we need to at first remove the cells from our cell culture media, and then we need to add around 0.4% of trypan blue, and we need to place in the hemocytometer chamber, and we need to calculate the viable cells. So now research problem 3, Mycobacterium tuberculosis H37RV is inhibiting interaction between phagosome, maturation, and now you have to design a suitable experiment to study the interaction of phagosome with lysosome. So again the same principle, you have to identify the phagosome and then you have to label the lysosome, and then step 3 you have to do fusion assay. And what is the observation you will get? You will observe the cells in the bright field and look for the bits on the cells. Observe the cells in fluorescence microscope with UV filter. If the bead has blue fluorescence, then the cells can be visualized through red channel. So the last topic is that what are the compositions of different organs? Blood consists of RBC, WBC, lymphocytes, neutrophil, basophils, eosinophils, platelets. Brain consists of neurons, astrocytes, glial cells. Liver consists of hepatocytes, Kupffer cells, blood. Heart consists of uh, cardiomyocytes, cardiac pacemaker cells, fibroblasts, smooth muscle cells. Cellular properties. So cell has different sizes, densities, different receptors on cell surface, and metabolic reactions. So these are some mammalian cells types, which are sperm cells, RBCs, lymphocyte, neutrophils, and so on. So they vary with their diameters. So we can use cell analyzer and differentiate different cells, differentiate between different cells according to their size, densities, fluorescence. uh, so the instrument is called FACS, fluorescence associated cell sorter. So the main components of flow cytometry is that the flow system or the fluidics, the optical system, and the electronic system or the signal processing. The flow system, the fluidics, central channel or core through which the sample is injected. Outer sheath contains faster flowing fluid, and then hydrodynamic focusing. The optical system consists of laser and arc lamp and argon lasers. So there are forward scatter channel. The intensity of the signal has been attributed to cell size, refractive index. Side scatter channel, the intensity of the signal is proportional to the amount of cytosolic structure in the cell. Flow cytometry, the electronic system or electronics. The electronic subsystem converts photons to photoelectrons. It measures amplitudes, area, and width of photoelectronic pulse. Also, it amplifies pulse either linearly or logarithmically, and then digitalizing the amplified pulse. So there are different types of plots: single color histogram, two color dot plot, two color control plot, and two color density plot. Then you have to do data acquisition, gating, selection of only a certain population of cells for analysis of a plot. Allow the ability to look at parameters specific to only that subset. Rectangular gets, elliptical gets, polygon gets, quadrant gets, histogram regions. So different fluorescent dye are there. FITC, whose emission wavelength is around 530 nanometer. Phycoerythrin 576, Texas Red 620, ECD 615, PC5 667, APC allophycocyanin around 660 nanometer. So by this you can observe apoptosis, cell cycle, DNA content, Ross generation, phagocytosis, oxygen burst, cytokine array. So different experiments you can do, at what stage of cell cycle cells are, you can measure. You can also do phagocytosis assay, cytokine array, you can sell, so Ross generation. So these are the different experiments you can do using this flow cytometry. In the next class we'll discuss in detail, more details. So today we'll be discussing now the week 9 assignments of previous year, so which will be more or less similar to this year. So if you have any doubt on this year's assignments, you can let me know. So the questions were, how many dichroic mirrors are present in an epifluorescence microscope? So we have studied it, only one dichroic mirror were there, right? So the answer will be 1. Kupffer cells are present in liver. In flow cytometry, the side scattered channel captures data related to cell granularity. In a cell cycle analysis by flow cytometry, the cells are stained with DNA binding dye, membrane specific dye, protein binding dye, or RNA specific dye. So it should be DNA binding dye, because in the cell cycle DNAs are getting replicated. So we can track the DNA, how much DNA content of the cell is. So we will stain with DNA binding dye. In apoptotic cells, annexin 5 binds to phosphatidylserine as phosphatidylserine in apoptotic cells translocate to extracellular side of plasma membrane. So this is plasma membrane. So phosphatidylserine becomes in the outside of, this is PS or phosphatidylserine. So we can track the phosphatidylserine. If it's outside, then it's apoptotic. Compensation is required in flow cytometry because what happens, spectral overlap between two fluorophores might happen. So that's why compensation is required. In flow cytometry, selection of data acquisition from a distinct fraction of cells from a cell population can be achieved by, in flow cytometry, selection of data acquisition from a distinct fraction of cells. So for that you need to do gating. Fixation of biological samples can be done by treating with what? Methanol. Permeabilization of fixed RBCs on a slide can be done by treating with 1% Triton X100. What is the purpose of this step? The purpose of this step to make the membrane more porous so that all macromolecules can enter into the cell. What would be the maximum resolution that can be achieved if a sample is illuminated by green light and observed through a oil emission objective with numerical aperture? So you have to calculate with that formula. So what was the formula was there? D equals to lambda by 2Na. Okay, so the resolution will be D equals to lambda, where it is around 514 divided by 2 into numerical aperture, that is 1.5, that is 3. Then 514 divided by 3, how much is this? It's around 171. So the answer will be 171. Philippine staining can be used to study the phagocytosis of latex beads in these specific cells. Philippine stains the cell membrane. Water in biological samples diffract electron rays and create noise. What it does, it diffract electron rays and create noise in cement image. Water is removed by dehydration steps. So it will be graded series of ethanol. Next, the coating of specimen with gold palladium is performed to increase sample charging on electron bombardment, make the specimen more conductive, decrease emission of secondary electrons, increase penetration of primary electrons. So the coating of specimen with gold palladium is performed to make the specimen more conductive. In TEM, The electron beam is focused on the specimen with the help of condenser lens. Malaria parasite culture is synchronized with the help of 5% sorbitol. So these are the some questions of week 9 content. So if you have any specific doubt, you can let me know. We have discussed week 7 assignments, we have discussed week 9 and week 9 content as well. So if you have any specific doubt, you can let me know. And next class will be coming with week 10 content, and then again the live assignments which are now live, will be discussing those. Else we will stop with today's class. Thanks for joining. Again we'll meet on next week from 6 p.m. 6 to 8 p.m. Thanks for joining.